Good morning and good afternoon, everyone. I would like to open the first PKU Blended Learning Journey 2025, which is a continuous medical education live webinar on advances in PKU management, dealing with current and future options. I'm Nenad Blau from the University Children's Hospital in Zurich, and we chair this webinar. And I have no potential conflict of interest. MEDEA is a medical education academy. It's a global medical education provider. It is dedicated to designing, delivering, and evaluating medical education programs worldwide, offering support to healthcare professionals in their continuing professional development. Trying to get the next slide. Yeah, here. In addition to the webinar that takes place today, there are a number of other informational options, different for postcasts, which took between June and October 2025. And today is the most important event, the webinar, which will start in a couple of minutes. I would like to introduce the scientific committee. There's the Kirsten Aring from Denmark, Amaya Belange-Quilintana from Spain, and Maria Grzewska from Poland, Anita McDonald from Birmingham in UK, Franz Ameo from France, and Julio Cesar Rocha from Portugal. And this independent educational program is made possible only through an independent educational sponsorship provided by PTC Therapeutics. Many thanks to them. The program today is divided in two sessions. The first session, starting right now, is dealing with the genetics of PKU, then the mechanisms of neurotoxicity of phenylalanine PKU with comorbidities in adult PKU, and then we will have a discussion. In the second part, session number 2, this will be discussed nutritional management of PKU, the women's health, including maternal PKU, new treatment options, and clinical trials on the market, and again followed by the discussion. This is the faculty. Again, many of them are advisory board members. In addition, we have Jerzy Giszewski, also from Poland, Carrie Harding from Portland, USA, and Anja Munter from Hamburg in Germany. The learning objectives of this webinar are understanding how variants in the PAH gene impact the severity of an individual's PKU, to recognize mechanism of neurotoxicity in PKU, to identify the need of personalized nutritional care with the use of adjunct drug treatment in PKU, pharmacological treatment, to evaluate selected aspects of health at different periods of life in women with PKU, and to identify new therapeutic options for patients with PKU and how to judge their clinical impact. Participants will earn 3 credit points for continuing medical education, which is certified by the CPD in Europe. And this will be achieved upon completing the evaluation form, and then you will get a certificate of attendance and the continuing medical education points. There will be questions and answers during the webinar, so please submit your questions during the lectures in the Q&A section, and faculty will address or answer your questions during the panel discussion at the end of each session, at the end of the first session and the end of the second session. The first talk will be given by Professor Anja Muta from the University Children's Hospital in Hamburg. She's the head of pediatrics there and has a long time experience with PKU. Anja, please.
Thank you, Nenad. Dear colleagues, dear friends, I was given the title Genetics in PKU from variants in the PAH gene to the PAH activity landscape. So this is my task for today, and I also tried to move. These are my disclosures. And the learning objectives for this talk are to understand the genetic heterogeneity of PKU, to understand how PAH genotypes impact PAH enzyme function, and to understand how visualization of PAH enzyme function can support patient counseling. What is the knowledge gap? Prediction of enzyme function from the genotype is challenging. Prediction of therapeutic options is also challenging. The comprehensive translation of genotype and functional data to the clinic is lacking, and comprehensive material for patients on genotype and enzyme function is lacking. Here, let's have an overview on the variants known in the PAH gene, and this is from the Human Gene Mutation Database. Here you find records for 1,078 different variants, and you see that we have exonic, intronic variants, but also small insertions, deletions, and so on. And the main message here is that missense mutations in the PAH gene are the most common variants that lead to PKU, with 60.6% of all PAH pathogenic variants and 70.6% of all pathogenic PAH alleles in PKU patients. Geographically, these mutations are not evenly distributed. And here you see that what we have collected for the PAH genotype and activity landscape database, that we looked at all published data, genetic data for different countries in the world. And one of the main things that you can see is that in the northeast, and this corresponds to Germany, Poland, Czech Republic, Slovakia. So I always hear Maria that says that in her country, and the same is true for ours, and even more in the Baltic countries, the mutation R408W, that is a very severe mutation, is more frequent in the northeast of the world. More lucky is Amaya, that because in Southwest Europe you find a lot of other variants that are milder and that come along with a higher percentage of BH4 response. In the US, R408W only 17%, a little like in Australia, and different genotypes in Latin America and China. So it depends where you live, and it depends, not where you live, but where you come from, what your genotype is likely to look like. The patient and the phenotype of the patient does not depend on single mutations, but on the genotypes. So in our database, we collected information from 10,895 patients. And the main message here is that 2/3 of patients are compound heterozygous. and we have a huge genetic variety. And only in 3% PAH, the PAH genotype is not conclusive. This can be because we only can identify one allele, and there are different genetic reasons for this, or we do not at all find a mutation in the PAH gene, and then we need to think about genes for BH4 deficiencies or in the DNA GC12 gene, for example. The PAH activity landscape of human PAH. What is this? This is an experimental way to mimic patient genotypes and to look at the resulting enzyme function of these genotypes. We used the cell culture models with a model with COS7 cells. We expressed enzyme from 2 different alleles, and then we analyzed PAH enzyme function at a broad range of phenylalanine and BH4 concentrations, reflecting different metabolic and therapeutic states. So it's important to understand that the PAH enzyme is extremely variable and can be regulated and works differently at different conditions. And this is important to understand. There is not the one enzyme activity related to one genotype. And this is the development of the data from rough to elegant. And first here you have the enzyme activity at varying Phe concentrations and BH4 concentration, and the whole thing is visualized by a color code. Blue, low activity, red, high activity. So here you see the raw data from a PAH activity assay performed in 96 well plates. And this creates a non-smoothed and non-modeled 3D surface. Here it's already a smoother variant of the landscape. This is the logarithmic transformation of the data, and then the exponential transformation leads to this nice smooth 3D and 2D model. But we will look at detail into the wild type PAH. So this is the healthy PAH enzyme at different Phe concentrations and at different BH4 concentrations. Very important, at very low Phe concentrations, so up to 80, the enzyme activity is inhibited. And this, of course, prevents depletion of the essential amino acid phenylalanine. With increasing phenylalanine concentration, the PAH enzyme activity increases to then decrease starting at around 1000 micromolar. So we call this substrate inhibition. And also with increasing BH4 concentrations, we have increasing enzyme activity, and this leads to this characteristic drop shape of the PAH activity landscape. Oh, excuse me. Can we go one back? Here you see an X, and this is the peak activity of the enzyme. So the best activity, the wild type enzyme works best at around 340 micromolar per liter phenylalanine and 150 micromolar BH4. And you see here the solid line, and this is 50% activity. and we call the region within this solid line, this is the optimal working range of the enzyme. So the idea is then, when we look at the genotypes later, is that for your patient, you find the coordinates that are linked to optimal enzyme function. So we expressed 42 different pathogenic variants, 16 common ones with allele frequencies more than 1%, and 26 rare variants. And this corresponds to 61% of all PAH pathogenic alleles. And we created landscapes for 99 different PAH patient genotypes that are known and described in patients. And this corresponds to 50% of all PKU patients that are registered in the PAH genotype and activity landscape database. The bio-PKU database that, thanks Ninat, has been built comprises 23,450 patient records with full genotype and phenotype data, 3,457 different variants with 4,688 different genotypes, and half of them are private genotypes. So only one patient so far is known to have this genotype. So this again shows the heterogeneity, and 70% are compound heterozygous genotypes. And we used this bio-PKU database for the genotype, for the 99 genotypes, and we retrieved the clinical phenotypes from 7,854 patient records and the BH4 response tests from 1,426 patient records. And by performing the landscapes, we identified different subpopulations. So subpopulation 0 with 18 genotypes, no residual activity, no BH4 response. Subpopulation 1, low residual activity, peak and working range is similar to the wild type, high rate of responders or slow responders, and the range of response is wide, so 24 to 100%. So we find different informations in the bio-PKU database. Subpopulation 2, low residual activity, 4 to 9. And we see a left shift of PAH activity, which means that the enzyme works better at lower Phe concentrations with a narrow working range. So you must bring the patient into this narrow working range at lower concentrations to give him optimal enzyme function. Mild clinical phenotypes, high response rate. In subpopulation 3, we see the other way around, a right shift of PAH activity at much higher. So it's really a difference here of PAH activity with a broad working range. So here it's easier to find a concentration of high activity, but more severe clinical phenotypes and 80% of BH4 response. So this group is challenging. Subpopulation 4, low residual activity, right shift, again, broad working range, severe clinical phenotypes, low rate of response. And this is the famous R261Q mutation. And these are the winners. High residual activity, mild phenotype, and they all respond. So here you see that we tried to show a visual distribution of subpopulations of PAH genotypes based on residual activity in 3 different dimensions, which is X, the phenylalanine concentrations, Y, the BH4 concentrations, and Z, so coming up, the residual enzyme activity. So SP1 has low activity, is at the bottom of the cage. SP2 has activity at lower Phe concentrations, and 3 and 4, red and orange, work better at higher Phe concentrations, but differ in enzyme activity. And SP5 have the highest enzyme activity of all subpopulations. If we compare the subpopulations as to maximum residual activity, we have these 4 groups that have lower enzyme activities, in particular 2 and 4, and a bit higher residual enzyme activity. When you reach 10%, it's already not so bad in 1 and 3. And Phe at peak activity, so we have these 2 that have lower, these 2 that work at lower Phe concentrations, 2 and 5, and we have these 2 subpopulations that work best at clearly higher Phe concentration, which is really a difference, and subpopulation 1 has a maximum activity close to the wild type. BH4 response, this is important for the patient. Subpopulation 0, no response at all. Then we have these 2 groups, subpopulation 1 and 4, where you see the variability is extremely high of BH4 response tests. And this may be because, not knowing the characteristics of the subpopulations, maybe the tests have been done at, let's say, unhelpful Phe concentrations or with different BH4 dosages. It is very clear that subpopulation 5 will always respond to BH4, and that subpopulation 2 also and 3 have a much higher probability to respond. For the clinical phenotypes, you have the far left, which has all classic PKU severe phenotype, and subpopulation 5, they have all mild phenotypes. But then you see there are 4 populations where you have all different phenotypes. And this again shows that this classification probably is not the right one, because we do not have 3 boxes, but we have a continuum of clinical phenotypes. So let's have a short look at the different classic representatives of the subpopulations. The R408W, subpopulation 0, the blue landscape. So if you see this, it's not good news for the patients. You know, Nenad always says the R408W does not have 0 residual activity. And this is true. It is 1 point something. Here it's, to make it easy, I said no residual activity. But the enzyme is aggregated, and for that not very functional. And we have severe phenotypes and no response to BH4, the blue landscape. Look at here. It's not so much different from the first one, but here it's there is a little green shadow, and residual activity is between 3 and 4. We have BH4 responders, mild phenotypes, and a good response to BH4. As soon as you see a little green light, things get better, and it gets even better here. We have, but you see the cross is clearly shifted to the left. So peak activity is here at maximum 105 micromolar per liter, micromolar. And you see that in this narrow range, you do find a considerable residual activity of 8%. And if you get the patient in here, he will respond to BH4 treatment, but not if he is at 800. This is the thing where you can use it for translational medicine. This one is a combined phenotype of subpopulation 3 with a lot of light green, but the maximum activity is very much shifted to the right to 575, 600 micromolar. And here, nothing works. If you push them down, they will have no enzyme activity at disposal. Frequently, they are severe, probably because they are pushed here and then protein tolerance is nothing, excuse me. And however, they have a high rate of BH4 response. Yes, probably if you move them to higher Phe concentrations. This is the very difficult R261Q that has a right shift of activity, and here a broad range of no activity until 300 micromolar. There is complete inhibition of the enzyme, and the BH4 loading test is challenging, some of you may know. And these are the winner: clear sky, sunshine, yellow, and all these patients have high residual activities, mild phenotypes, and a guaranteed response to BH4. This is the publication from this year. Paulina, read it. We published this many years ago, and we refined the method and the analysis with and so on. And this is the paper you can look up. And we would like to conclude that PAH activity landscapes are derived from a cell-based model that detects residual PAH function across a range of Phe and BH4 concentrations and mimics human genotypes. The clinical phenotypes can be predicted and treatment regimens can be adjusted based on residual PAH function profiles. And the graphical representation using a color code is a helpful tool for patient counseling. I would like to invite you to visit our website, where you can look at different countries, where you can search for genotypes, and where you also can request genotypes. We now again have personnel to help you with genotypes and landscapes on demand, and then you will receive a report with clinical advice that comes from me. Thank you very much for your attention.
Thank you very much, Anya, for this excellent presentation. Now, I will ask my colleague, Amaya, to continue with the next.
Thank you, Nena. Thank you, Anya. My name is Amaya Bellinger Quintana, and I'm the head of the metabolic department in one of the reference centers in Spain, in Madrid. First of all, I would like to remind you all to put your questions in the question and answer or the chat options you have underneath, so we can answer them at the end of this session. And now it's my great pleasure to introduce Professor Maria Gwisaska, who is the head of the pediatric department in the Pomeranian Medical School in Szczecin, Poland. And she's going to give us a very comprehensive presentation regarding neurotoxicity in PKU. So thank you, Maria.
Thank you, Amaya. Thank you for media for organization of this webinar. Well, if I can change the slides. Okay, these are my disclosures, maybe. Okay, so this is the subject of my presentation, which is mechanisms of multidirectional phenylalanine neurotoxicity. These are my faculty disclosure, and what about the learning objectives of this presentation? It is to understand the key mechanisms responsible for the multidirectional neurotoxicity of phenylalanine, to identify modern diagnostic methods that assess the consequences of high blood Phe levels on the brain, and to be able to consider the importance of maintaining low blood Phe concentration for the optimal functioning of the central nervous system. While talking about phenylalanine neurotoxicity, I always like to start with the picture of Øyvind Felling, the person who discovered, the physician from Norway who discovered PKU in 1934. Felling was the first who linked intellectual disability presented by two small children coming from Egeland family with a huge amount of excretion of phenylpyruvic acid in the urine of this children. So two things were demonstrated: severe intellectual disability and excretion of huge amount of phenylpyruvic acid. In the coming years, other scientists were really interested in understanding what is going on in the brain of patients with PKU, and in 30s and 40s, many of them perform detailed studies of individuals with severe intellectual disabilities. Most of these patients came from the special institution dedicated to them, and among all these scientists, I would like to underline the great contribution of George Jervis and Lionel Penrose. And so in 30s and 40s, the term oligophrenia phenylpyruvica was introduced, which later on was changed into phenylketonuria. And both of the scientists were deeply involved in the discovery of the nature of PKU. Penrose, additionally, was one of the first who started to consider the possibility of reducing phenylalanine intake as the treatment option, and he was also the one who established an autosomal recessive pattern of PKU inheritance. And already in 30s and 40s, it was clear that PKU, which at that time was totally untreatable disease, was responsible for one of the most severe forms of intellectual disability. But we now know that despite severe intellectual disability, these patients may present with severe drug-resistant epilepsy, with the whole spectrum of psychotic behaviors, including destructiveness, self-injury behaviors, agoraphobia, and many others symptoms. Many of them have severe speech difficulties, and many of them, especially later in life, may present severe neurological presentation. Nowadays, we also know that even early treated patients, especially when they are young children or even in older age, when they are losing metabolic control, can show the whole spectrum of psychological and psychiatric problems, including, especially in younger children who are not properly treated, decrease in IQ, but also in other patients, attentional problems, school problems, and many of other symptoms which may significantly impair the quality of life of these patients and their whole families. So really, PKU is very challenging disease. We know that especially the young children's brains are sensitive to high levels of phenylalanine, but now we know that the brain remains sensitive to neurotoxic influence of phenylalanine throughout the whole life of the patients. And this is a very nice graph coming from one of the paper from 2013, showing that the higher are Phe levels, especially during childhood, the higher is probability that these children and also older patients will reach at the end the IQ below 85. So this is a strict correlation between the IQ and the blood Phe levels. And in 30s and 40s, when scientists had only limited numbers of equipment available, using mainly the eyes and the microscope, they were performing number of studies trying to understand the neuropathological findings in the brains of PKU patients. And this is a very nice example of some later paper. This is a paper by Baumann and Kamper, who again performing the study on the brains of untreated PKU patients who were doing the morphologic and histoanatomic evaluation of these brains, and despite the morphologic changes in the form of reducing the brain size and the way how the brain tissue was developed, there were numbers of problems related to myelin status in the brains of these patients. But additionally, what you can see on the right part of this slide, you can see that they very nicely show the differences between the morphology of single neural cells between PKU patients and healthy control. And as you can see that in PKU individuals, neural cells are smaller and with the smaller, lower numbers of dendrites. Therefore, in such a situation, creation of network between the neural cells is significantly limited. So now we know that the whole brain may be affected by high levels of phenylalanine. Therefore, neuropathologic changes are present both in white matter and in cerebral cortex. In white matter, it is mainly hypomyelination and astrogliosis, demyelination with status spongiosus, progressive white matter degeneration. Regarding the cerebral cortex pathology, mainly we have to deal with the decreased gray matter volume, especially in posterior brain, developmental delay with reduced nerve cell size, and arrest in migration of cortical neurons. Okay. Okay, we know, let me. There were also some animal studies performed on the PKU mouse hippocampal cultures treated with phenylalanine, and these studies showed the reduction in synaptic density and dendritic length, some structural alteration, and reduction in dendritic arborization and malformation of cortical dendritic trees. So again, when there is a reduction in dendritic arborization, there is a reduce in the ability to create a proper network of different cells. And there is also a decrease in synaptic pruning, which is the process aimed to clean the brain tissue from unused and synapses. And we also know that there are some studies mainly coming from 2012 and some coming from some late years later from Adler Abramovich and Ehud Gazit, colleagues from Israel, who underline the possibility of amyloid etiology in PKU. And they explain to us that that in PKU, amyloid fibrils, which were found and which are formed by phenylalanine, have ability to bind to lipid membrane and are known to induce apoptotic cytotoxic effects and apoptotic cell death in dose-dependent manner. And so if all these things are going on in the brain of our patients, we have to ask the question, how it is possible that the phenylalanine can crosses the blood-brain barrier. Well, this is possible thanks to the existence in blood-brain barrier of one very particular transporter, which is called LAT1, and this transporter is dedicated to transport from the blood to the brain all neutral amino acids with the highest affinity to phenylalanine. Here you have the list of these large neutral amino acids, and please notice that that to this group belong tyrosine and tryptophan, which are absolutely essential precursor for the synthesis of serotonin and dopamine. But the other large neutral amino acids are essential for the proper synthesis of the brain proteins. Therefore, as you can see on this graph, and here this large neutral amino acids is marked in violet color, that in the case if the patient has high blood Phe levels, phenylalanine has a simple ability to block this transporter for the transport of other large neutral amino acids from the blood to the brain. As a result, in the brain, there are the huge amount of phenylalanine, which additionally has an ability to decrease the activity of some enzymes involved in the synthesis of serotonin and dopamine from tryptophan and tyrosine. So we can ask, what are the results of elevated Phe levels in the blood? Well, most of all, they are related to increased brain Phe levels and decreased brain levels of large neutral amino acids other than phenylalanine. So it is not only a story about phenylalanine in the brain, but also a story about the consequences of lack of other large neutral amino acids transport from the blood to the brain. And it was found that in mild PKU, even with not so high levels of blood phenylalanine with the status of so-called supraphysiological plasma Phe concentration between 200 up to 500 micromole per liter, there is a significant reduced transport of tyrosine and tryptophan across the blood-brain barrier because phenylalanine is simply blocking this transport, which result, and also additionally, the reduced cerebral protein synthesis, which is not good because we know that protein synthesis is needed for proper cognition. So even in early and continuously treated PKU patients, if they are with even slightly elevated Phe levels, there may be some impairment in the proper transport of large neutral amino acids from the blood to the brain. And this competition is sometimes even more important than high Phe levels in the brain by itself, because it can cause the situation which is named cellular undernutrition. It means that not sufficient amount of this large neutral amino acids is transported from the blood to the brain, causing reduction in the incorporation of these amino acids into the brain proteins. It can also affect altered DNA methylation with gene dysregulation. So this may be another cause of problems in the brain. It can cause reduction in the number of polyribosomes, reduction in the cells number, and altogether, this is one of the reasons why many of untreated PKU patients have microcephaly as the result of reduction in brain weight. But this kind of things that I just mentioned do not explain myelin abnormalities, which are very well known among our patients with PKU. There is no doubt that proper synthesis and maintenance of myelin is essential because myelin and axon form a functional unit. They have to connect to each other. And we know that myelin axonal interaction transduces signals that upregulate the production of two very important enzymes that I just mentioned, tyrosine and tryptophan hydroxylases, which are involved in the conversion of tyrosine to dopamine and tryptophan to serotonin. So we also have to remember, we have to remember that myelin is produced by oligodendrocytes, which are glial cells in the central nervous system that assemble and maintain myelin. And there are some subpopulations of oligodendrocytes, especially in the posterior brains, but also in other parts of the brain, that they are extremely sensitive to phenylalanine. And if they are exposed to phenylalanine, they are just simply losing the ability to produce myelin. And this is a nice graph coming from quite a historical paper already. And on the top, you can see the graph of normal brain or the brain of PKU patients properly treated, where oligodendrocytes being not exposed to high levels of phenylalanine show nice ability to produce myelin, which covers the axon. And then when myelin and axon are in close contact, everything is nicely produced, including neurotransmitters. While below you have the PKU brain, where oligodendrocytes exposed to high brain Phe levels lost their ability to produce myelin, therefore axon is not covered by myelin, and the production of neurotransmitters at the end of the synapses is significantly impaired. And these myelin problems in PKU patients may be partially related to negative influence of phenylalanine on the activity of the enzyme, which is called 3-hydroxy-3-methylbutyryl coenzyme A reductase. And this is quite a key enzyme involved in the metabolism of cholesterol. And we all know that cholesterol is essential for the production of myelin. And this myelin changes found in the PKU patients are described as the white matter abnormalities, and they are a result of two possible situations: or demyelination, which is equal to loss of already formed myelin in treated individuals when they lost the metabolic control, or hypomyelination. It is the situation where there is a lack of myelin formation in untreated individuals. And we know that these white matter changes are partially reversible in the situation when even untreated patients can reach lower Phe levels. Then again, oligodendrocytes are starting their ability to form myelin. And as you can see in one of my patients, when he was totally on very high Phe levels, the extent of this white matter changes was quite significant, and it's markedly it improved when we were able to keep this patient for the period of 9 months on the lower Phe levels. And now let's come to the way how we can even more demonstrate all what is going on in the brain of our patients. So there are numbers of studies nowadays showing different negative influence on of high Phe levels on the brains of our PKU patients. Here you have a presentation showing the correlation between the impairment of white matter and its correlation with lower cognitive performance in adults with PKU, and other papers dedicated to this subject. Moment. Mhm. Mhm. Oh, sorry. and this is a more advanced neuroradiological method, diffusion kurtosing imaging, which detects even subclinical white matter abnormalities in patients with phenylketonuria. And there were also numbers of studies showing dedicated to morphometric analysis of gray matter integrity in individuals with heavily treated PKU, and it was shown that there was a correlation between Phe concentration in the blood and with the decreased gray matter volume in cortex and even in cerebellum. And in cortex, these changes were seen especially in posterior brain, but co-authors concluded that there is, however, there is a link with the blood Phe levels, and these changes, more data are needed. And some years ago, the group of Professor Fritz Treffs with Andrea Pilotto and their colleagues performed a very elegant study on early treated adults with phenylketonuria, and they found that there was a strict correlation between the Phe levels and the concentration of neurotransmitters metabolism metabolites in cerebral spinal fluids. Moreover, they were able to correlate this metabolites, as you can see here, with some degree of gray matter atrophy in particular region of the brain. So in summary, based on the volumetric studies, it was found that there is a reduction in global white matter volume as well as reduction in the volume of several subcortical gray matter structures. So we can summarize that the whole brain, all the parts of the brain are sensitive to high brain Phe levels. Just recently, Professor Fritz Treffs with his group published some studies showing us that there may be also some problems with the glucose transport via blood-brain barrier. You can see here the concentration of glucose in the brain in comparison with GLUT1 patients and with the control. So with PKU, in PKU patients, this glucose concentration is somewhere in the middle, and it was found that low glucose level can disrupt the oligodendrocyte function and myelination. So for sure more studies are needed on this subject. And there are also information about the negative connections between high blood and brain Phe levels and antioxidant defense, because phenylalanine can negatively inhibit the activity of several enzymes involved in antioxidant defense, as well as can show negative influence on the activity of number of enzymes involved in energy production in the brain. And we all know that the brain is the tissue and organ which requires a lot of energy for the proper functioning. So the brain of our PKU patients still remain a mystery, and there are many question marks. We know what we are sure that there are many of different processes going on which may be negatively or the other way impaired by the metabolic consequences of PKU. And it was all these theories were some years ago very nicely shown in the paper by Professor Frank-Jan van Stronsen and co-authors, where they nicely presented that it is not only the story related to high phenylalanine in the brain as the results of the high blood Phe levels, but also the problems related to the disturbances in the proper large neutral amino acids transportation from the blood to the brain, which may negatively impact not only the protein synthesis, but the whole metabolism of neurotransmitters, and many other processes may be significantly impact while in the situation of high brain Phe levels. Therefore, there are still a number of questions that are not solved. So there are some take-home messages from my presentations. I would like to underline that phenylalanine is neurotoxic. There is no doubt that it is harmful, that its harmful effects are multidirectional and involve both structural and functional brain alteration, that even in moderately elevated blood Phe levels, there can be some degree of impairment in large neutral amino acid transport, brain protein synthesis, myelination, neurotransmitter production, and brain energy metabolism. And we know that white matter and synaptic development show high vulnerability, especially during postnatal brain maturation, so in younger children. And keeping blood phenylalanine as close to physiological or as low as possible can support optimal cognitive and neuropsychological functioning in PKU. Therefore, I strongly believe, and I think I hope that you will agree with me, that optimal metabolic control can result in optimal brain protection in PKU. And thank you very much for your attention.
Thank you, Maria.
Yeah.
We need to forward to the next presentation. Discussion will be at the end of the session, as you already know. And the next presentation will be on, can I have the first slide, please? Yeah, the next presentation will be on comorbidities in adults with PKU and will be given by Professor François Mayo. He is a professor of internal medicine at the University of Tours, France, and he is the head of Department of Internal Medicine, which is part of Reference Center for Inborn Errors of Metabolism at the University Hospital of Tours. And he is involved mainly in the management of adult patients with different inherited metabolic disorders, including PKU. Please, François.
Thank you very much, Nenad. So it's difficult to talk after these two brilliant ladies, but I will do my best. My presentation will be clinical about you have the title here, the comorbidities in adults with PKU. Can I have the next slide? I don't have the control. Yes. So here are my disclosures. Next slide. Sorry. No. Yes. So now, can we go back, please? Sorry about this slide problem. Okay. So the learning objective of my presentation is very simple, is to understand the range of comorbidities in adult patients with PKU. And my talk will be focused only on early treated PKU, meaning people who are screened, who are diagnosed through the newborn screening of PKU, as we have in many countries. So we all agree that PKU is a success story from the identification of the disease, as Maria presented to you by Professor Folling, through the diagnostic methods, the implementation of newborn screening in many countries to effective therapies. And we see now that we have more and more treatments for PKU, which is good news. However, although we have all these success stories, adults with early treated PKU are still considered by experts to have suboptimal outcomes, and in the form of complications and comorbidities. And if we look at this paper by Greg Hens in 15 years ago, you have on the right side here the long list of suboptimal outcomes about adults with PKU. And I will try to show you among this very long list what is the reality of this suboptimal outcome. In the literature, there is somewhat a confusion in most papers between what we can call complications and comorbidities. So I would like to make it clear that complications refer to conditions of symptoms that arise as a direct consequence of the disease itself or but is inadequate treatment. That means that these complications are directly related to hyperphenylalaninemia or the Phe-restricted diet, because we know that this Phe-restrictive diet is able to control high Phe levels, but there are also some potential complications of the diet itself. Regarding this complication, I will go quite quickly through that point. We have data from case series and case report, mainly about neurological complications of the disease in adults. And I will show you just a very small part of a study we have conducted in France, which is the ECOFEN cohort study. When we talk about comorbidities, there are other medical conditions that occur more frequently in patients with PKU than in the general population, but they are not necessarily caused directly by the disease. We saw this, I will show you these comorbidities, but the link between hyperphenylalaninemia and these comorbidities is not so clear to date. This data that I will show you comes from health insurance claims and one for a systematic literature review that I will explain. Just some word about the neurological complications. What we see is subtle deficits in neurocognitive functioning, and this is mostly about the executive functions or attention, and they are quite mild in this group of patients, and the relations to Phe levels is still quite unclear. In some other patients, but it represents a very small group of patients, we can observe some and reversible somatic complication. I will insist of this reversibility because these complications like tremor, but more severe ones like spastic paraparesis or vision loss due to occipital leukoencephalopathy, they are rare. They occur in case of poor metabolic control in adults who has a completely relaxed diet, and they are reversible if we are able to decrease the phenylalanine level in these patients by resuming a proper phenylalanine control diet. In the ECOFEN study, which was a national prospective cohort study, which is closed now, it was funding by a national program for clinical research that we have in France. So this is this is a public funding. What we have done, we have included 191 patients, and 4 were excluded for different reasons. And at the end, we were able to analyze 187 patients, including 105 52 classic PKU patients. And it was not a surprise because it is we have more classical type of patients in our country due to the genetic background. The patients we had at baselines, we have more women. The mean age was around 30, so it was a young population, but they were all screened when they were newborns. Almost half of them were still treated, so means that half were not no more treated with phenylalanine restricted diet or Kuvan for some of them. And you see that, of course, the serum phenylalanine at inclusion was the higher in the classic PKU group if you compare to the other groups of patients. What we saw most is we saw a rate of neurological complications of 11%, exactly 10.7%, so in 20 patients. And what was interesting is that we saw this complication only in the classical PKU group, which is the most severe of the disease. Regarding these neurological complications, it was quite heterogeneous, and it's the same that I described you previously from the literature, like tremor, spastic paraparesis, or vision loss. So now I will move to the other part of the presentation, which are the comorbidities. So we have in the literature so far 3 different papers with the same methodology, meaning that the data come from health insurance claims. One was published in the USA, one in France, and one in Germany. So I will start with the American data first, the paper published by Barbara Burton in 2018. And I will show you the main findings. It's interesting because they were able to analyze more than 3,500 patients and compare to 18,000 controls non-PKU patients. And what they found an increase prevalence, and this is based on the reimbursement of medication of these patients, that they have a twofold risk of kidney failure with hypertension, that they found a higher rate of overweight in these patients. They found also that a higher risk for kidney failure without hypertension, some digestive problems like gastritis and esophagitis, and some other problem like osteoporosis. Osteoporosis, it's not really can be considered a comorbidities, but more as a complication of maybe PKU, but more probably to the phenylalanine restricted diet, and some other findings that are more difficult to understand. For example, a higher risk for asthma, dermatitis, or gallbladder disease. Maybe gallbladder disease may be also related to a higher risk of weight, of obesity. We don't know. When we move to the French study with this methodology, I was involved in this experiment, in this paper. We tried to be more selective regarding the population, because in the US population, there were also some late treated patients included in the analysis of the data. So what we did here from the French health insurance database, we have isolated early diagnosed PKU patients. So, and you see at the bottom of the slide, we were able to get the data from 1,500 patients that we compare two controls. So at the end, we were able to compare these patients to 7,500 healthy controls. You see again that the mean age was quite low, around 30. And what we found most is we found a higher rate of hypertension again in these patients as compared to the non-PKU control. We found no difference regarding diabetes. We found a higher rate. You see the PKU patients are in blue in this graph. We found a higher rate of chronic renal disease, although we don't know if this is related to hypertension or to another cause. No difference in cholesteremia. Osteoporosis, I told you maybe it's more a complication, but we found more. And we found no ischemic heart disease at all, 0 events in this population with early treated PKU. I insist on this because there was maybe could be a confusion from the other data I will present you now. And now we'll move to the German study published by our friend Friedrich Treffs from the German health insurance claims. And what they found also, so you see there are some similarities between these three studies, although there are also some differences. The group of patients they were able to analyze was smaller if we compare to the French and the US study, with a little bit less than 400 patients, and they compared to almost 4,000 non-PKU patients. And what they found a higher rate of, again, overweight or obesity, and they found a higher rate of chronic ischemic heart disease. And the main difference here for me is because they have also they were not able to separate early or late treated in this population that they are analyzed. And for me, with one of my students, we look very carefully at all the paper published on this topic. And for us, this is very clear that the higher risk for chronic ischemic disease is for the late treated PKU population and not for the early treated PKU population. They also find higher risk for unspecified diabetes mellitus. We don't know if diabetes is type 1, type 2, or whatever. A higher rate of lipoprotein metabolism that we didn't find. Again, as the American study, a higher rate for asthma and some other problem related, again, you see here again, some digestive issues and also dermatitis, menopausal or other perimenopausal disease. I have no details about, but there was a higher prevalence. From this systematic literature review that we have done with some of the experts who are here today at this webinar, we found some other things, but what I would like to stress from this, I will not go into details into the methodology of the paper, but we found that people, we calculate like a burden score of comorbidities, and we found that this score was higher in Phe-restricted diet group patients as compared to healthy controls. So showing overall that the adults with PKU, they have a higher comorbidity burden than non-PKU population at the same age. So this is what I would say like a confirmation of or a synthesis of the data I showed you in the previous slide. There is something also very interesting published in 2022 by Barbara Burton again. It's like a survey among experts of PKU to know which comorbidities they should require special attention in adolescents and young adults in PKU. And you see, it's like a mirror to the data that I showed you before. These experts, they agree at 100% that we should look at the weight of the adult patient with PKU, to screen for overweight or obesity. We should also be careful regarding the risk of type 2 diabetes, but I think also this is related, of course, to overweight of obesity, gastrointestinal disorder, renal disease, asthma. And you know, this is not a surprise because this comes from the literature that the experts have examined. Also allergies, osteopenia, ischemic heart disease, and also nutritional deficiencies. So my message to finish is that these patients with early treated PKU, they may develop, this is not 100%, complications due to high phenylalanine levels, like neurological or maybe bone disease or some other minor complications, but they may also encounter some comorbidities as adults. And it's not recommendations or guidelines, but I really think that we need to implement a long-term follow-up of this patient. And this was in the European guidelines that we updated this year, that we published this year. Then this long-term follow-up will make it possible to screen for neurological complications. And to my opinion, it relies first on a physical examination, make a yearly or more frequently if needed, neurological examination to this patient first before doing any MRI or whatever. Listen to the patient, examine them to screen for this complication. We should screen for overweight or obesity to provide this patient a proper nutritional management. For sure, I am really convinced from the data I show you, we have to screen for hypertension. So I would recommend to check blood pressure at every visit of these patients. It should be part of the physical examination of every adult PKU patient. And also screen for kidney failure by dosing plasma creatinine. I put a question mark at the frequency, but if I do remember, we recommend to measure plasma creatinine once a year in the European guidelines. I think, yes, this is interesting to do. So thank you for your attention and ready for questions.
Well, thank you, Francois, for your great presentation. Now we're opening the discussion to the whole panel. So please, if you have not done so before, either write your questions in the question and answer or the chat, and we will tell the different panelists, or raise your hand and the technicians will alert us and you can do your question directly. So Nenad, I think you had something to ask.
Yeah, I think we have already a couple of questions. The first one from myself to Professor Muntau. What would be the predicted or observed landscape for homozygous R4AW with sepiapterin?
We need to do this. And this is
What is your
exactly,
prediction?
I don't know. So from what we see in clinics, we should see a little green light. But this is
more-
exactly what we now want to do, is that we will do the whole panel with previous treatment of the cells with sepiapterin. We are planning the project because, yes, this will show the differences between BH4 and sepiapterin. But I hope, and is what we've experienced in patients, that we will see at least some % of response.
Thank you. There is another question by Kerry Harding. Please remind us what the typical BH4 concentration in the liver is likely to be when we are considering the PAH landscape. Is it known how much liver BH4 increases after a sapropterin or sepiapterin treatment?
Carry. So the concentration in the liver is, if I remember, what, 25 to 50 micromolar, and it increases by 10 to 100 fold with treatment.
Thank you.
Well,
And I have just one more from my side to Professor Mayo. What are the comorbidities in PKU patients with cancer and under anti-cancer drugs?
You mean, is there something about cancer in PKU?
Yeah, it could be that a PKU patient suffer from cancer, and which is some kind of comorbidities, right?
It is.
Yeah,
So
severe one.
yes. So there are two parts. In one of the studies, there is a little signal for cancer, that maybe there is an increased risk for cancer. And I know some other data from the French health insurance, that there is also the same over risk of cancer in patients with PKU. And there are some works right now. There is a team working on the link with phenylalanine and cancer. So maybe there is something, but this is the beginning of the things. Regarding your question, your question is difficult. Your question is difficult. A PKU patient with cancer, I would say treat cancer first, but
Sure.
yeah. If it's any, when I had it's not cancer, of course, but I had a patient, a PKU patient with severe burns. And while he had all these, while they were reconstructing all the skin with all the grafts and so on, he required like a loading test of phenylalanine because he needed it for skin growth. And afterwards, of course, when all the grafts were there, he actually went into hypophy, and he was a child, so we were a little bit worried because of how much the Phe was being used for skin growth. And maybe in cancer, we might see something like that maybe the cancer cells use phenylalanine, and maybe Phe levels could change because of the cell growth. So it should be something that maybe alerts us. If suddenly a PKU patient is overly well managed, it might not be a good sign. So it was something to keep in mind.
yes, that reminds me that Anita MacDonald has published a paper that we have participated some years ago like about the management of patients with PKU and other disease. And there were some cases of breast cancer, if I remember.
You know, Francois, I participated in the study. I was just about to join this discussion with one of my, at that time, 5 years or 6 years old boy with classical PKU, homozygote for 400AW, who developed acute lymphoblastic leukemia. And of course, as you said, and this case was included in this presentation, in this publication. And of course, as you said, the first was to treat leukemia, which was very difficult because especially in the beginning phase of this treatment, there is the huge amount of steroids that you are given. So on steroids, his Phe levels, even if we were trying as much as we could, were up to the sky. It was very difficult, and after a certain period of time, he was transplanted and now is doing just fine. But this treatment of leukemia was very challenging because then, as the side effect of chemotherapy, he had numbers of severe changes in his mouth. His appetite was decreased. It was very challenging, very challenging. But of course, we had to concentrate and focus on leukemia treatment, and with the transplant, the problem was solved.
Yeah, I raised this question because it is known that the treatment with methotrexate and some other anti-cancer drug inhibit the dihydroteridine reductase and induces biopterin deficiency, in addition to existing phenylalanine hydroxylase deficiency. So there's from two sides, the patient is loaded with additional phenylalanine. But I can see that Carrie Harding is raising her hand. Carrie, please.
I was just going to add to that discussion. I don't know about the specific use of the, specific chemotherapeutic agents. I've just found in the couple of cases I've been involved in that the catabolic state of the patient, and then when they become ill from the chemotherapy, it's just been impossible to keep their Phe levels under control, and that becomes a secondary issue. I don't know what I would do if it was somebody with MSUD or an organic acidemia. It'd probably be a lethal problem. I don't know how. I haven't had to face that. I don't know how I would manage it. PKU, you just sort of, in my case, it's just been give up and let the Phe levels go where they may be. But it's been really difficult.
Mm-hmm.
Yeah, interesting. There are no questions from the participants. Amaya, what should we do?
Well, it's 3 o'clock, and I know some of the panelists and so on are. We need to finish on time. We have we have like 5 minutes. I have one question for Maria, and but we would like the participants to make their own questions as well. And so please don't hesitate even now. Raise your hand like Carrie did. But for Maria. I know you've talked a lot about how all these neurotoxicity is not just Phe levels. It's like the nutritional, the brain nutrition. So and how important it is for patients basically to have access to treatments, to new treatments, even at any age, even if they're late diagnosed. What do you have to say about that?
Well, I strongly believe that, first of all, low phenylalanine diet saved the brains of thousands of PKU patients. There is no doubt about this. But I am more and more convinced that the lower, closer to physiological Phe levels are, the better may be the outcome of these patients, because I am not aware about any particular protecting mechanism which may protect the brains of our PKU patients against high Phe levels. And I very like from this point, we've heard this several times when Carrie Harding said that, you know, patient with diabetes mellitus has to be kept in the strict glycemic control, and I strongly believe that the same is with PKU patients. And I think, coming back to your question, that for example, adults with untreated PKU, implementation of this kind of pharmacological treatment may be very beneficial, because even late introduction of treatment, so far it was mainly low phenylalanine diet, but we also know that Kirsten in Denmark is treating untreated adults with large neutral amino acids. Anyway, decreasing the Phe levels in the brain of untreated patients may show significant benefit in the quality of life of these patients. Maybe not to all of them, but many of them should be given such a chance, and of course in cooperation with the families, with the caretakers, but they can improve. It is very difficult at the beginning to predict who will answer to this kind of intervention, but many of them should have been given this kind of chance. So I strongly believe that even untreated patients, adults, should be for them, we should introduce a trial of any kind of treatment.
Thank you, Maria. Before the coffee break, we have time for two short questions. The first one from Melanie Hill to Professor Mayo. What is your opinion of risk of developing dementia in later life of patients with PKU?
This is, I think, the biggest questions that we all have in mind regarding the future of our early treated PKU, adult PKU population. So what I just can say now, we don't know. Maria, I think it's also related to the presentation of Maria, showing this toxicity. And we know now that there is also a toxicity in adults. You showed the problems of the amyloid deposit in the brain of mice and also humans with PKU. So, because now they are only, for example, in France, we start at the national level. The newborn screening started in 1972. So now they are like 53. So we don't know. We will see within the next years if we have a higher rate of dementias compared to the general population. But that's the big question.
Yeah. Thank you, Francois. And I believe there are more people with dementia without PKU than with PKU. Right. Okay. Now we have a coffee break, and we will get paid back at 15:20.
Thank you all. Please be on time, because as I say, we have to be on schedule. Thank you.
Session of today's webinar focusing on nutritional management, women's health, emerging treatment options in PKU. So we are proud to present a group of distinguished speakers who will share their expertise on these important topics. So let's begin. And the first speaker to introduce in this session is my friend and colleague for 30 years, Dr. Anita MacDonald. She is a consultant dietitian in inherited metabolic disorders at Birmingham Children's Hospital and an honorary professor in dietetics at Plymouth University in the UK. Although she is semi-retired 9 years ago, she even more involved in PKU work concentration on solely on this group, as well as doing some voluntary work for the National Society for PKU, NSPKU. Anita's involvement in inherited metabolic disorders has spanned over 5 decades. Dr. McDonald obtained her PhD in PKU in 1999. She has directly cared for more than 500 patients with PKU. She has always been actively involved in PKU research, supervises PhD students, master students, and lectured worldwide on PKU topics. Anita has more than 500 publications. Many are research publications on PKU. She is an active member of the European PKU Guideline Group, a member of ESPKU's Scientific Advisory Committee, as well as a member of the UK NSPKU Medical Dietetic Advisory Panel and Scientific Advisory Committee. So the title of her talk will be Advances in Nutritional Management of PKU. Please, Anita, the floor is yours.
Thank you very much, Madam Chairman, and thank you very much to Flaminia for your very kind invitation today. These are my disclosures, and these are the learning objectives today. So we're going to identify the need for personalized nutritional care with the use of pharmaceutical drug treatment in PKU, and we're going to explore new advances in nutritional support in PKU. So let's first talk about pharmaceutical treatments. With pharmaceutical therapy, some individuals may achieve an unrestricted protein intake without the need for low-Phe protein substitutes, whereas others still require substantial protein restriction. Generally, it's expected that drug treatment should increase natural protein intake by at least 100%, unless baseline intake is already higher than safe levels of protein intake. When patients start drug therapy, it's important to counsel patients extensively about changes in dietary treatment. Individuals need to learn how to reprogram and develop good eating habits. They need to know what healthy protein is, that protein just doesn't come out of McDonald's or Kentucky Fried Chicken or the Pizza Hut. They will need help in meal planning, education about food preparation and food budgeting, and we need to be able to address any nutritional gaps that are a consequence of changes in nutritional intake. When natural protein is increased, this process should be approached with caution. It is recommended to use a stepwise approach. For example, firstly, low protein foods such as low protein bread and pasta can be replaced with regular food sources. Then foods such as egg and cheese could be introduced into the diet, and finally followed by meat and fish. But of course, this has got to include regular monitoring of dietary intake, looking at the food patterns, looking at weight gain and the body mass index, as well as also checking for the blood phenylalanine control. Now, as natural protein increases, the protein substitute intake should be lowered in parallel by the same amount of protein equivalent. In practice, reduction in protein substitute intake is commonly delayed by professionals. A systematic review and meta-analysis of 306 people with PKU who were receiving long-term sapropterin treatment found that only half of them stopped taking low-Phe protein substitutes. Reducing the intake of low-Phe protein substitutes may result in a reduction in micronutrients, particularly vitamin D, folic acid, iron, calcium, iodine, and zinc. And also, when we're looking at protein substitute reduction for our patients, we've got to consider the impact of illness, because we know with drugs such as sapropterin and sepiapterin that patients will still have peaks in blood phenylalanine levels at the same time as illness, and patients need the help of protein substitutes to help control blood phenylalanine levels. We've got to consider the efficacy of drug therapy with age. We may find a 1-year-old will tolerate 20 grams of protein with drug therapy. That is great. They can have many normal foods. But it may well be that the rate of protein increase doesn't increase at the same rate. And at the age of 4 or 5, they may only be tolerating 25 or 30 grams of protein intake. So need to rely more on taking protein substitute. We've also got to think about the possibility if the drugs may be stopped for some reason, and then we've got to reintroduce the protein substitutes. And we also have to think about situations when there are increased protein needs, such as patients with increased physical activity, with growth during pregnancy. So all of these things are always in the back of our mind when we're looking at reduction in protein substitute. If I can go back 1 slide, please. 2 slides now. Sorry, I went on too fast. 2 slides back. That's it. Greater dietary freedom may result in a higher consumption of higher energy meals and snacks. To prevent obesity, it's important to develop satiety awareness. We can do many simple things. For some patients, when they go on to drug therapy, so often in the past, they've been used to eating small packets of everything. That's all they've been allowed to have. Suddenly, when they go on to drug therapy, they can have more protein. Then they can go on to large packets of snacks, and patients don't know when to stop. So we've got to help them with information about portion control. Encouraging behavior strategies like mindful eating encourage individuals to slow down and enjoy the food. Filling the plate with extra vegetables to increase volume without extra calories, all of these things will help. And of course, increasing exercise. There are still many information gaps that we don't know about nutrition with drug therapy. We've got to do the work to find out. We don't really know how long-term protein tolerance differs between the different drug therapies, particularly with some of the newer drug therapies. We don't know much about long-term growth, body composition, and a rate of obesity with the drug therapies. We also don't know much about nutritional status, including bone mineral density. We don't really know if eating patterns and disordered eating improves with time with drug therapies. There's been no controlled studies. And what about the impact of drug therapy on blood phenylalanine stability when children are under the age of 2? We may have children tolerating extra protein, but it may be at the cost of loss of control because some of the drug therapies are not very good during illness, and children under the age of 2 have frequent illnesses. Then what about the impact of erratic drug administration, its impact on control and nutrition, and the quality of nutritional intake of women on drug therapy in pregnancy? All of these things we need to research. All of these things we need to get much more information on to help us with directing our nutritional therapy. Then innovations in protein substitute formulations, the palatability and nutritional profiles offer new opportunities to improve diet quality and patient experience. Physiomimic technology is an engineered delivery system. It's a multi-step granulation and coating process that has produced a prolonged release protein substitute composed of phenylalanine-free amino acids coated with ethyl cellulose and sodium alginate. This fiber-based matrix enables gradual gastrointestinal release, replicating the absorption kinetics of intact protein such as casein. Unlike conventional free amino acids, substitutes which are rapidly absorbed and cause a sharp plasma peak increase followed by declines. Prolonged release protein substitutes promotes a slower, steadier release that stabilizes plasma amino acid profiles. A randomized controlled crossover study in children with classical PKU showed that a once daily evening dose of a prolonged release protein substitute significantly reduced early morning blood phenylalanine levels with less variability following overnight fasting. This controlled absorption also improved metabolic control and improved tyrosine availability. In this randomized crossover study in ARMOR A, participants received the prolonged release amino acids as a single evening dose for 7 days, followed by a washout period, then crossed over to receive the usual amino acid protein substitute for all the daily doses for another 7 days. In Arm B, the order was reversed. Administration of the prolonged release amino acids significantly reduced early morning phenylalanine levels compared to amino acids that weren't coated only. Prolonged release amino acids also significantly increased blood tyrosine levels and improved the Ty-Phe ratio. Now, one of the big problems with the slow release protein substitutes right now is that the formulations aren't always the easiest to get into patients. They're not the most appetizing or palatable, but I truly believe this kind of technology has got legs, and I think it's something that our manufacturers can work with and improve in the future, and will certainly help patients with classical PKU. And then we come on to casein glycomacropeptide, or GMP. This, as many people know, is a bioactive peptide derived from cheese whey, and it's an alternative protein source for patients with PKU. The downside is that it does contain some phenylalanine, around 36 milligrams for every 20 grams of protein equivalent. Now, we have a lot of work right now to show that in humans with PKU that it will improve palatability. It is associated with a better gastrointestinal tolerance. It does improve microbiota. It does improve breath odor. It is associated with reduced blood phenylalanine variability. It's associated with improved body composition and higher blood tyrosine. However, because of the phenylalanine content, we know very little about the use of GMP in general, whether the phenylalanine content will put people off from prescribing this for their patients with PKU. And as a consequence of this, Sharon Morris did a global questionnaire looking at the use of GMP with dietitians. Now, this questionnaire was sent to all our colleagues in the world. Sharon got 208 responses from health professionals from 43 countries. And I think many of you who are listening this seminar today took part and actually answered the questionnaire. So we're very grateful to you, and thank you very much for this. Now, what she got from the responses, that from the 208 responses, the professionals cared for something like 14,000 patients with PKU. And around about 25% of those patients were being prescribed GMP as a source of the protein substitute. Interestingly, most of these patients weren't being given the GMP as a first-line protein substitute. They was being given the GMP after trying amino acids, and this had failed for some reason, maybe because the patients didn't like it. Also, very interestingly, only about a quarter of those patients were being given up to 50% of their protein substitute requirements from GMP. Very few of these patients were actually getting the full protein requirement, the full protein substitute equivalent requirement from GMP. Now, it's often suggested that the absorption of GMP is slower than amino acids. And two of our researchers, Anne Daly and Alex Pinto, looked at the absorption of GMP. They studied 20 healthy adult volunteers, and the absorption of GMP was compared with phenylalanine-free amino acids and casein. So what did they find? Well, what they found was that the amino acids from GMP were not absorbed more slowly than the phenylalanine-free amino acids, and were certainly a lot more rapidly absorbed than casein, which is a known slow-release protein. This applied to total amino acids, branched-chain amino acids, large neutral amino acids, and essential amino acids. The only thing where benefit was shown was with tyrosine. This showed a gradual increase over 240 minutes in contrast to amino acids and casein. So this was more slowly absorbed. And we've also seen with GMP that blood tyrosine levels are higher. We're not quite sure why this happens, but it does. Now, precision fermentation is an emerging biotechnological approach that utilizes engineered or evolved microorganisms to sustainably produce high-value proteins and other functional ingredients. Through advanced strain engineering, microbial cell factories are programmed to synthesize a target compound, such as a specific amino acid or peptide, with a controlled fermentation environment. Following production, the desired ingredient is separated and purified from the host microorganism. This technology holds significant promise for the development of protein substitutes suitable for PKU and other metabolic disorders as well. But it certainly offers the potential to create phenylalanine-free formulation with tailored nutritional profiles and improve sensory characteristics. And then we move to low protein foods. And one of the options here that we have to explore is the 3D printing of low protein foods. How does this work? Firstly, we design a low protein food model. We come up with the idea. We create a recipe for it. We then make up that recipe, create an edible paste. This is put into a cartridge. This is then put into a machine that will build the food in layers. And then we get a final food that is beautifully created, but is a printed food. Now, the advantages of 3D printing is we can produce any food, any shape, using any ingredients and any nutrient profile that we desire. And you can be really quite creative with this. Many top chefs use this kind of technology already. It's associated with less waste, and also it's already being used in the world of therapeutic dietetics. It's being used to produce meals for patients with dysphagia. So it's something that's not new to us as dietitians. However, there are some disadvantages with this technique. The texture is limited. The machinery is relatively high cost, but not as expensive as some of the drug therapies. The processing time is slow, and you need quite a lot of skills in order to operate it. At the minute, these factors hinder its scalability and mainstream adoption, but it's got a load of possibilities. And certainly we need to scale up this technology to see if we can improve the range of low protein foods available for our patients with PKU. And finally, I just want to mention artificial intelligence. This has got the ability, if I can go back, this has got the ability to really transform our world in the management of diet and PKU. Artificial intelligence will enable meal planning platforms. We can create sophisticated algorithms. We already know that there's papers reported trying to calculate phenol intolerance of patients with their genetic profile using artificial intelligence. It's got the ability for us to combine many of our global databases looking at the phenylalanine content and amino acid of foods. Really, AI is our oyster as far as developing what we do, and certainly can make our workload much easier. So take-home messages for today: continued research and advancements in nutritional therapy will enhance our ability to manage PKU and hopefully make life and much easier for our patients. But always careful and ongoing monitoring will remain essential for our patients. Thank you.
Thank you very much, Dr. Anita McDonald, for this excellent presentation. And I'm sure there will be some questions for you, but we will move on to the next speaker. And that will be Dr. Yuri Jezewski from Szczecin in Poland. So it's my pleasure to welcome him. He's a specialist in obstetrics and gynecology. And for many years, he has served as a senior consultant and faculty member at the Department of Gynecology and Obstetrics in Pomeranian Medical University in Szczecin. With decades of clinical and academic experience, he has developed particular expertise in gynecological endocrinology, infertility diagnosis and treatment, antenatal care, and advanced gynecological and obstetric ultrasonography. His professional focus also includes comprehensive care for women during the menopausal transition and in older age. He contributed as an external expert to the European guidelines in PKU from 2017, offering valuable insights into female reproductive health in the context of PKU. Dr. Jezewski is a frequent speaker at patients' meetings dedicated to the care of women with PKU. His lecture addressed the key issue across the female lifespan, including puberty, reproductive planning, pregnancy, postpartum care, and menopause. His work continues to bridge gynecology and metabolic medicine, advancing care for women with rare inherited disorders, especially PKU. So please, Dr. Jezewski, the floor is yours.
Thank you, Kirsten, for the very kind introduction, and thank you to media for invitation to this webinar. As gynecologist, I feel honored to have been invited among such distinguished group of specialists. Ladies and gentlemen, dear colleagues, in my presentation, I will discuss issues related to the health of women with PKU at various stages of their lives from gynecologist perspective. And I have no conflict of interest to disclose. And what are learning objectives of my presentation is to appreciate the role of gynecologists in the PKU metabolic therapeutic team, to understand the leading function of ovaries at different stages of women's life, to recognize the need of special gynecological attention at pivotal moments in women's life, and last not least, to raise gynecologists' awareness of the specific needs of women with PKU and the ways in which they can help address them. So, globally, worldwide, we have nearly 500,000 of patients with PKU, and approximately 50% of them are women at different ages. This therefore presents a substantial area of opportunity for gynecologists. Women with PKU in different stages of life need professional gynecological counseling for puberty care and fertility control in reproductive maturity, for pregnancy planning and pregnancy care, for ensuring the satisfactory quality of life during adulthood, menopause, and the late climacteric age. So when discussing the role of gynecologists in teams of specialists treating PKU patients, we must ask ourselves whether we, as gynecologists, are aware of the existence of women with congenital metabolic disorders in our daily work. Do we know who and where in our region manages metabolic disorders? What do we gynecologists need to know about phenylketonuria? The most important thing to know for us gynecologists is how excess phenylalanine affects the patient. If untreated or inadequately treated, PKU can result in central nerve system impairment. Low Phe levels can save the brain of the treated woman. Even more important is to know how toxic excess of phenylalanine could be to the fetus of pregnant women with PKU, leading to maternal PKU syndrome. Metabolically prepared pregnancy can save the fetus. A woman with PKU should first be seen as a woman, not only as a PKU patient. Which physiological factor has the greatest impact on women's life? Sex hormones, particular estradiol. They are primary physiological regulators of everything, of female development, higher intellectual function, reproduction, bone structure, and cardiovascular health, metabolism, mood, and sexual function throughout the lifespan. So the steroid secretion fluctuates throughout a woman's life. Estrogenic activity rise steadily beginning at puberty. Thereafter, mature ovarian cycles produce marked cyclic fluctuations in hormone levels that cause a range of symptoms varying by cycle phase. During pregnancy, the hormonal profile becomes relatively stable and rather pregnancy-related symptoms predominate. And finally, when ovarian estrogen production declines markedly in peri and postmenopause stages, numerous vasomotor and other deficiency symptoms occur, leading to sustained reduction in quality of life. On this chart, I would like to present the phases of women's life. First, the childhood. Period of hormonal calm. Then, young girls reach the onset of pubertal maturation marked by the first menstruation, followed by the reproductive maturity phase. Despite young age, from the first period onwards, we must take into account the reproductive readiness of a young girl. The purple curve, illustrating the reproductive activity of ovaries, climbs rapidly and reaches the plateau and slowly declines. This stage of decline of ovarian function started around 35 years of age is called reproductive aging. Then we move to the period preceding the last menstruation in women's life, which is called menopause. This term means only the last bleeding, after which there were no bleedings for 12 months. So, period for 5 years before menopause is known as the perimenopause, and 3 to 5 years after menopause is a postmenopausal period. The more general term climacterium with climacteric symptoms begins with perimenopause and lasts until the end of life. All periods of women's life are determined by the reproductive activity of ovaries, for which estrogen secretion is secondary. The reproductive function of the ovaries is assessed by measuring the concentration of AMH, anti-Müllerian hormone, a key parameter of ovarian reproductive reserve. And here, very important notes. The first one, menarche, first menstruation in women's life, is preceded by a full ovarian cycle with a fertile phase during which pregnancy is possible. And second, a woman's fertility is possible until menopause. All this information are crucial for understanding the difference between particular stages of women's life. Thus, at the end of relative hormonal silence of childhood, hormonal activity begins in adolescent girls, starting with the first ovarian cycle, which ends with the first menstruation. Does this have clinical significance in phenylketonuria? In recent 2025 Orphanet publication, Horka and co-authors reported the increase in phenylalanine concentration toward the end of the menstrual bleeding and lower phenylalanine level after ovulation at the beginning of luteal phase. The difference observed were not sufficiently large to warrant dietary changes. Around the time of menarche, the gynecologist's role is to plan with the parents of young PKU girl the first gynecological visit, which should occur after the first menstruation. Particular attention should be paid to girls who may be sexually active even before menarche. European PKU recommendation from 2017 state that from the age of 12 years, all women should receive systematic age-related sex education with professional counseling about the risk of unprotected sexual contacts. They should be informed that unplanned pregnancy can occur even during the first menstrual cycle. The gynecologist must recognize situations requiring the use of fertility control methods. Fertility control methods, what the methods consist of? They prevent pregnancy in sexually active women. The effectiveness of these methods is measured by many methods, for example, the PER index. The PER index is a measure of contraceptive efficacy indicating the number of unintended pregnancy that occur per 100 women during 100 years of use for a specific birth control method. A lower PER index indicates greater effectiveness of contraceptive method. So we are talking about fertility control in women with PKU due to the consequences of metabolically unprepared pregnancy. Only the most effective methods of fertility control should be considered. And the most effective methods are those referred as a long-acting reversible contraception. Other available methods are significantly less effective. Let us remember that the consequence of poorly chosen contraception will be an unplanned pregnancy and the risk of maternal PKU syndrome. So, what are the key points within reproductive maturity stage? The most effective birth control methods should be individually tailored. Effective contraception may be only discontinued after attempting to conceive after metabolic preparation to achieve sufficiently low Phe levels. And we shouldn't forget about the women with a Phe concentration between 360 to 600 micromole per liter who are not treated. So, regular preventive visits during reproductive maturity should include counseling or family planning, education about the concept of maternal PKU syndrome as a worst-case scenario for women with PKU during their fertile stage, underlines the principles of collaboration with the whole therapeutic team when preparing for pregnancy, educate for the need of avoidance of risky sexual behavior and substance use, advising on preventive vaccination. Oh, I've done something on my computer. Okay. Sorry for the break. And we are moving to the next stage of women's life. We can call it married, working, wish to plan pregnancy, or we can call it family planning. And women with PKU can give birth to a healthy child. It is absolutely possible. Specialist team collaboration is essential. Transition from contraception stage to pregnancy planning is only possible when Phe levels are between 120 to 360 micromole per liter. And we have to remember that if the pregnancy of women with PKU is not properly planned and managed, the fetus may be irreversibly damaged by phenylalanine, which acts as a multi-organ teratogen. The critical period of impact of high Phe concentration on fetal central nerve system, cranium, and heart development is 5 to 8 weeks of gestation, counting from the first day of last menstruation. But telling the truth, on the real duration of pregnancy, it is 3rd to 6th week of the pregnancy. So many women may not know yet that they are pregnant. So symptoms of maternal phenylketonuria syndrome observed during pregnancy can be detected by ecologists during routine obstetric visits. The hemorrhage in uterine cavity that increase risk of miscarriage, of the lack of fetal heart rate, intrauterine growth retardation, microcephaly, esophageal atresia with a very characteristic ultrasound picture, polyhydramnion, congenital heart defects, facial dysmorphia as in fetal alcohol syndrome, neurological problems and abnormal muscle tone, which can be observed in low fetal biophysical profile, structural organ malformation in liver, on kidneys. The effect of uncontrolled maternal PKU are the same regardless of whether the fetus has PKU or not. European guidelines on phenylketonuria contain very accurate statement that maternal PKU is a high-risk pregnancy. This term opens further proceeding with pregnant women with PKU as a key, which opens the algorithm of management of high-risk pregnancy. So this pregnancy should be followed by obstetrician who is well informed about PKU, as well as with metabolic physician and metabolic dietitian. Optimal birth outcome occurs when maternal blood Phe levels are between 120 to 360 micromole per liter and are achieved minimum 2 weeks before conceiving and maintained throughout pregnancy. Phe control should be done once per week during pregnancy planning and twice a week during whole pregnancy. Detailed follow-up with ultrasound examination is specific for higher risk pregnancy with the ultrasound screening and target prenatal imaging. In the case of unplanned pregnancy, emergency treatment procedure, immediate metabolic consultation to reduce Phe concentration. But what if there is no pregnancy? World Health Organization defined this problem as a failure to achieve a pregnancy after 12 months or more. Infertility affects millions and may affect also PKU families. So in complete European guideline on phenylketonuria, there is a statement that as maternal PKU treatment needs considerable effort from women and her partner, failure to conceive requires special attention. And therefore, it is reasonable to refer well-controlled patient of fertility specialists earlier than after 1 year. Referral to fertility centers should be done after 6 months of unprotected sexual activity. But very important remark, if the partners have a history indicating significant fertility limitations, the preferred approach is even to forego the 6 months waiting period and instead initiate diagnostic and therapeutic workup for both partners as a first step in attempting conception. And another issue. Many studies on egg and sperm quality conducted in vitro fertilization programs after several failures have shown that the various metabolic substances, including high concentration of phenylalanine, have a negative impact on the process of gametogenesis. For this reason, if possible, it is advisable to extend the period of maintaining low phenylalanine concentration up to 90 days, the duration of gametogenesis, before discontinuing contraception. So when all our patients have dozens of children and they don't want to be pregnant again, we have the stage of reproductive aging and perimenopause. But what we have to have in mind during these stages is that pregnancy is still possible, even with climacteric symptoms. So highly effective contraception methods should be recommended during this stage. And then we have climacterium, a period of women's life associated with progressive ovarian failure and with the depletion of ovarian reproductive reserves. The climacteric period accounts for approximately 1/3 of women's lifespan. Around 80,000 PKU women worldwide are currently in the climacteric phase. In countries where newborn screening for metabolic disease was implemented many years ago, women with early diagnosed phenylketonuria are now reaching menopause. Climacteric stage is characterized with numerous of symptoms. And the symptoms could be also as a result of some metabolic disorders. Here we have common symptoms for climacteric period and high Phe levels in women with PKU. Mood changes, cognitive complaints, fatigue and decreased energy, headaches, hair and skin changes, changes in body odor, osteoporosis, and increased fracture risk. So, should women with PKU receive special gynecological care during climacterium? Yes. First of all, we must identify such a patient through the thorough medical history. If there are no contraindications, come up with proposal of hormone therapy for climacteric period. Hormone therapy will significantly improve quality of life. And what is very important here, due to some common climacteric symptoms, and high phenylalanine concentration. The evaluation of the effect of hormonal therapy should be performed together with a metabolic team. So, during this turbulent period of changes in women's body resulting from the climacteric period, phenylalanine concentrations and varying quality of life, therapeutic success can only be achieved through the cooperation with a team of specialists. And appropriate hormonal and metabolic balance in women with PKU during climacterium ensure a high quality of life. So in conclusion, what are the take-home messages? What should be considered in gynecological care in women with PKU? We should pay particular attention to young girls who may be sexually active before their first menstruation. In women with PKU, we should use only the most effective methods of contraception. Effective contraception should be discontinued, only attempting to conceive after metabolic preparation and achieve sufficiently low Phe levels. Maternal PKU is considered as a high-risk pregnancy, so we should follow the special algorithm for high-risk pregnancies. In climacteric age, important is to identify women with PKU and metabolic consultation before starting hormone therapy. So, from the gynecologist's perspective, a PKU woman's life from puberty to beyond menopause can be lived to the fullest with a friendly help from gynecologists as a member of therapeutic team. And well-prepared and well-controlled pregnancy is the most beautiful gift a woman with PKU can give to her future child. Thank you for your attention.
Thank you so much, Dr. Kiszewski, for this very interesting and very relevant presentation, since all of us have a growing adult PKU population with women who gets older and a lot of potential mothers who want to have a healthy child. So thank you so much. I'm sure there will be questions, but for the sake of time, we will save them for the panel discussion. Thank you.
Hello, and it should be now my time to introduce our next speaker. Hello, so it is my great pleasure to present our next speaker, which is Professor Carrie Harding, who is a professor of molecular and medical genetics and pediatrics at Oregon Health and Science University in Portland, in the USA. We know that Professor Harding is a fantastic physician dedicated to patients with all kinds of inborn errors of metabolism, particularly phenylketonuria. He is also a true passionate of science, deeply involved in developing new therapeutic approaches for inborn errors of metabolism, including PKU. He has been and continues to be one of the leading investigators of pharmacological treatments for this disorder. In his laboratory, he also conducts advanced research on gene therapy in PKU. Moreover, Professor Harding is an excellent lecturer, actively participating in numerous scientific conferences. And today we have a pleasure of listening to his presentation on new treatment options and clinical trials in PKU. So, Carrie, the virtual floor is yours. Can you hear me? Hi, Carrie.
Carrie, you can start.
Yeah, sorry, for some reason my microphone all of a sudden had a problem. Can everybody hear me?
Yes, we can. Yes, we
Okay.
can. So
Sorry about that. I don't know what happened there. But thank you. I'm pleased to provide today a summary of pharmacologic treatment options that are currently available for PKU, and also to talk a bit about a potential future treatment option that is in clinical trial.
This is my faculty disclosures. These are my learning objectives for the day. So these include understanding the current unmet treatment need in PKU, learning about currently approved pharmaceutical treatments for PKU that may lessen dependence upon a fee-restricted diet, and finally to understand a bit about promising new therapy, which is inhibition of the SLC 6A19 large neutral amino acid transporter in kidneys, which induces urinary Phe excretion. The mainstay, the mainstay of contemporary therapy for PKU, of course, continues to be dietary phenylalanine restriction. And the PKU diet certainly has been the paradigm of success in preventing severe cognitive disability associated with untreated hyperphenylalaninemia. But significant shortcomings in dietary treatment remain, however, in my opinion, and this drives the need for continued development of novel therapies that lessen the dependence upon dietary Phe restriction. Maintenance of low blood Phe, of course, is recommended for life, but the complexity and the unpalatability of the Phe-restricted diet complicates strict adherence to the diet for many individuals, and particularly for adolescents and adults. And this becomes very difficult for them to maintain adherence to the diet. And particularly when their Phe levels go up, they become afflicted with a brain fog that can cause significant neurocognitive deficits and psychiatric symptoms that I've listed here. And we've already heard about emerging comorbidities and complications and the maternal PKU syndrome. So these are my personal perspectives on the current treatment goals of PKU treatment, and whether the goal is 360 micromolar, which is the current recommendation in the US for adults, or even the 600 micromolar, which is the recommendation in the European guidelines. In my opinion, it's really impossible to achieve those goals if dietary Phe restriction is the only available treatment. There are several concerns regarding the long-term use of dietary intact protein restriction and free amino acid supplements. And we don't fully understand the possible consequences of moderately elevated blood Phe over the course of the lifetime. And why should we accept blood Phe of 360 when the normal level is between 35 and 85? And I think Maria already mentioned my thought of why do we accept 6 times normal in PKU when we don't like to have our diabetics being 6 times normal? Why should it be okay for persons with PKU to have 6 times normal Phe levels? So in my opinion, if we could do it safely, why not keep blood Phe within the normal physiologic range? And at the same time, if we could, normalize dietary intact protein intake. So there are many different potential therapies for hyperphenylalaninemia. I'm only going to focus on 3 particularly. I'm going to talk about methods for supplementing tetrahydrobiopterin, or BH4, which is the required cofactor for phenylalanine hydroxylase. We'll talk about enzyme substitution therapy with phenylalanine ammonia lyase, or PAL. And then finally, we'll talk about the SLC 6A19 inhibitor. So phenylalanine hydroxylase, or PAH, of course, catalyzes the conversion of phenylalanine or Phe to tyrosine, but the reaction requires the pterin cofactor tetrahydrobiopterin, or BH4. And BH4 is synthesized de novo in liver from GTP, but the BH4 supply is predominantly maintained through enzymatic recycling. So in individuals with hyperphenylalaninemia, BH4 synthesis is actually upregulated already, and the concentration of BH4 in liver is elevated when you're hyperphenylalanemic. But Professor Shigeo Kure in Japan and his colleagues first observed that in a subset of individuals with PAH deficiency, Blood Phe concentrations will decrease if the person's given pharmacologic doses of BH4 orally. And as has already been discussed, only individuals with particular genotypes are likely to respond to BH4, and that's likely because the mechanism of action is that BH4 is acting as a molecular chaperone to stabilize a partially functional PAH enzyme. So only individuals with PAH genotypes that yield some minimal residual enzyme activity are likely to respond to treatment. And those with severe variants that lack any activity whatsoever will likely not respond to BH4 treatment. So cypropirin dihydrochloride was the first approved pharmaceutical for the treatment of PKU, which gained FDA approval in the US back in 2007 and worldwide approval within the following year or so. Cypropirin is a synthetic form of BH4, and it's supplied either as pills to be taken by mouth or as powder to be mixed into liquid and drunken. They're both available as brand name, and then there are 2 generic suppliers as well. So the safety and efficacy of cypropirin was evaluated in a series of clinical trials that culminated in the phase 3 PKU-003 trial, which demonstrated a significant difference in the mean blood Phe of individuals taking cypropirin 20 milligrams per kilo per day in comparison to those taking placebo in a group of participants who'd already been determined to be responsive to cypropirin treatment. The long-term safety and efficacy of cypropirin treatment has been tracked in a couple of registries, one in the US known as the PKU registry, which includes data on 1,159 individuals with PAH deficiency who have been exposed to cypropirin at some point. And of those, 504 were found to be responsive and have maintained continuous exposure. So the mean blood Phe concentrations and their dietary Phe intake are depicted here in this chart in blue. The curves in red are data from individuals who only had short-term cypropirin exposure and are likely to have been non-responders. So the mean blood Phe of the continuously treated cohort decreased by 32% from 591 to 392 micromolar after 5 years. And that was associated with an increase in mean dietary Phe tolerance from approximately 1,000 milligrams at baseline up to 1,530 milligrams at 5 years. Now, sepiapterin is the most recent addition to the pharmaceutical toolbox for the treatment of PKU. It's a precursor of BH4 that's provided orally, and it gained marketing approval in Europe and the US just earlier this year. It's approved for use in individuals with pH deficiency over 1 month of age and who have documented blood Phe concentrations over 360 micromolar. So sepiapterin is a naturally occurring precursor of BH4, and it's actively transported into cells and then converted to BH4 via the salvage pathway. There are data suggesting that sepiapterin has increased bioavailability over cypropirin. Cypropirin, which is fully reduced BH4, does not readily cross cell membranes. Sepiapterin crosses cell membranes unchanged, including the blood-brain barrier, results in high intracellular concentrations, and it's then converted to BH4 intracellularly, and thereby enhances the metabolism of phenylalanine to tyrosine. It also supports production of dopamine and serotonin in liver, acting as essential cofactor, in brain, acting as the essential cofactor for tyrosine and tryptophan hydroxylases. The safety and efficacy of sepiapterin has been studied in several clinical trials, which included the pivotal phase 3 Affinity trial. And here, 157 participants of all ages were enrolled and were challenged with sepiapterin. So blood Phe concentrations decreased at least 30% in 103, or 66% of the study cohort. 110 individuals in the total cohort were randomized to either active drug or placebo. And after 6 weeks of treatment, mean blood Phe had decreased 63%. Sorry, I have to go back. So that's over here on the right. 63% they had a mean decrease of 63% in their blood Phe. from baseline in the sepiapterin cohort in comparison to the placebo group. And the mean blood Phe of the sepiapterin group had decreased down to 236 micromolar, which is below the treatment targets of both the US and the European guidelines. So this study also included individuals who were treated with cyproterone before they switched to sepiapterin. And as you can see here, the blood Phe concentrations were somewhat lower on sepiapterin than they were on cyproterone in this population without any change in their diet therapy. So attempts to increase dietary intact protein have been carried out in this extension phase of the trial, and this has reportedly been successful, but these data had not yet been formally published. We'll now turn to enzyme substitution therapy with pegvaliase. Pegvaliase is recombinantly produced phenylalanine ammonia lyase, or PAL enzyme, from the blue-green algae Anabaena variabilis. The enzyme is heavily coated with polyethylene glycol, or PEG, in order to modulate the immune response against this foreign protein. Following subcutaneous injection, pegvaliase circulates in the bloodstream and it deaminates phenylalanine to transenamic acid. And cinnamic acid is further metabolized into hippuric acid in the liver, and then that's excreted in the urine. And pegvaliase is supplied in pre-filled syringes in three sizes, three available sizes of 2.5, 10, or 20 milligrams. The safety and efficacy of pegvaliase has been studied in a series of phase 1, 2, 3, and now some post-approval trials. The pivotal efficacy data was provided by the phase 3 PRISM trial, which was a blinded, placebo-controlled, randomized discontinuation trial. So we couldn't run a typical placebo-controlled trial because if you initiated a placebo control at the outset, because everyone develops antibodies against pegvaliase, and most people get hypersensitivity reactions, at least a skin reaction to the injection. So we would have immediately known who was on active drug and would be unblinded to who was on active drug. So to do a properly blinded study, we had to start everybody on drug at the beginning, wait until they'd had a treatment response, and then take some people off and put them on placebo. So these data show their blood Phes at the beginning of the trial before anyone was on treatment. And then this was, they were all started on medication, and then this was after they'd shown an initial response. They actually dropped further later on after they've had further exposure to the drug. But at this point, a third of them were randomized to go on to placebo. And then 8 weeks later, you can see the difference between the blood Phes in the individuals who went on placebo versus those who remained on active drug. There's obviously a large, significant difference between the two, and this was the data that proved the efficacy of Phe lowering with pegvaliase. This, to me, though, is the most powerful data from the study, which is the long-term extension of continuous exposure to the drug. This is data out to 3 years, but we have data out to 5 years, which was recently published. And what's shown here is the mean blood Phes in all of the patients on all doses. There's 261 total individuals exposed here, and the doses include anywhere from 5 milligrams 3 times per week up to individuals who needed 60 milligrams every day, and this is dependent upon their immune response to the drug. So some patients need very little and others need quite a bit, depending upon how many antibodies they need. And what's shown here is their decrease in blood Phe in red, down to a mean of just a little over 300 micromolar. What's not depicted here is that more than half of the individuals have mean blood Phes less than 120 micromolar. And at the same time, what's shown is that their dietary phenylalanine intake has more than doubled over that period of time. In the cohort of individuals who had ADHD symptoms, which was not all of them, but those who did have ADHD, their ADHD symptoms decreased over time, and that decrease in ADHD symptoms correlated with their decrease in mean blood Phe. As I've already mentioned, everyone who's treated with pegvaliase produces antibodies against the drug. They produce both anti-PEG and anti-PAL antibodies, and these antibodies are most prevalent in the first 6 months of treatment. They begin to wane with continued exposure. The antibodies are responsible both for hypersensitivity reactions, which are mediated by complement fixing type 3 antigen antibody hypersensitivity. And those antibodies also ultimately determine what dose will ultimately be necessary to achieve efficacy in any individual. Erythema and swelling around injection sites, as well as arthralgia, are the most common reactions to pegvaliase, as shown in this table. We've developed a risk mitigation strategy that helps to lower the risk of severe hypersensitivity events. First, all patients receive antihistamine pre-medication prior to injection, at least for the first 6 months of treatment, and any time that dosing is being escalated. And patients and an independent observer are trained to recognize the symptoms of a potentially severe anaphylactic reaction and to administer an epinephrine autoinjector if necessary to prevent a serious reaction. A typical induction titration schedule has been designed that begins with a small injection of 2.5 milligrams once per week and then slowly increases the frequency and size of pegvaliase dose to minimize the chances of a severe hypersensitivity reaction. And about half of patients are able to complete the schedule without interruption, and they achieve 20 milligram daily dosing within about 9 weeks. However, we readily slow this schedule down if a patient begins to have significant hypersensitivity, and I'll often back down to a dose that gives them a minimal to no reaction, and then continue with that dose through about 24 weeks when the antibodies start to mature. And then we know then that their reactions will be much less, and we can attempt to dose escalate after that point. So about half of the patients require prolonged titration to ultimately achieve efficacy. So I want to illustrate the potential for pegvaliase through this example. This is a 33-year-old male PKU patient of mine who at baseline was consuming about 70 grams of intact protein, which was clearly too much, and he was taking a large neutral amino acid supplement. Because of the elevated Phe, he suffered from inattention that interfered with his ability to maintain employment, and he also had a lot of psychiatric symptoms. And clearly, as was displayed in this plot, his blood Phe was very elevated when we initiated the pegvaliase. Because of the elevated blood Phe, he suffered from inattention that was interfering with his work. He had almost no reaction to the drug and completed an induction and titration in the expected 9-week time frame. And once he reached 20 milligram daily dose, his blood Phe plometed. And I've added a plot here that shrunk down the y-axis so that you can really see the detail in his Phes, and which really dropped to nearly undetectable on 20 milligrams. And unlike PAH and liver, which is allosterically regulated and will shut itself off, pegvaliase does not have allosteric regulation, and it's just on all the time. So the more pegvaliase you have circulating in the blood, the more Phe it consumes. And that's why Phe can drop below normal. And during the phase 3 trials, we had more than 100 subjects with below normal blood Phe for as long as 18 months without any apparent adverse effects. But remember, this is in the context of them consuming normal amounts of dietary protein. So I really wasn't worried about the fact that he had low blood Phe, but I did ultimately decrease his dose back to 10 milligrams per day, and his blood levels have settled nicely back into the normal range. The best thing is that in association with the improved blood Phe control is that his neuropsychiatric symptoms have completely resolved. This is our current cohort of pegvaliase-treated individuals at our clinic at Oregon Health and Science University in Portland. And this is people we've treated post-approval in 2018. I've had experience with treating other individuals in the trials. We have 3 patients who are still titrating up and haven't gotten to efficacy currently. We have had 13 patients who've discontinued due to adverse events, and that's been their choice to stop the drug. But we do have 38 patients who've achieved efficacy with at least a 20% decrease in their blood Phe. And I have 33 who maintain blood Phes less than 360. 23 with less than 120, and all 33 of those patients have been able to normalize their, completely normalize their diet. So the final treatment I want to discuss is that's in a novel that's currently in clinical trial is the pharmacologic inhibition of the large neutral amino acid transporter SLC6A19, which is a transporter in kidney that transports all of the large neutral amino acids from the renal filtrate back into the plasma. And if you inhibit this in individuals with hyperphenylalaninemia, it preferentially causes the increase of phenylalanine out into the urine. And inherited loss of function variants in SL6A19 causes generalized large neutral amino aciduria in a phenotype known as Hartnup disorder. And that's a trait that typically causes no symptoms. JNT517 is a small molecule allosteric inhibitor of SLC6A19. And that drug causes amino aciduria when taken orally. And that's depicted here. And JNT517 has now been studied in several phase 1 and 2 clinical trials, including multiple phase 1 single and multiple ascending dose studies in healthy adult volunteers, and in a phase 2 randomized double-blinded placebo-controlled dose study in adults with PAH deficiency. It's been well tolerated with minimal adverse effects. And an oral administration of the drug has been associated with dose-dependent large neutral amino aciduria. And impressively, pH deficient adults who received the highest JNT517 dose of 150 milligrams twice per day have exhibited a mean 60% reduction in blood Phe at 28 days into the trial, which is the longest exposure that anyone has had. Importantly, the plasma concentrations of the other large neutral amino acids, which are also affected by SLC6A19 inhibition, were not decreased below normal in either the healthy adult volunteers or in PAH deficient adults. And based upon these initial positive data, there are longer-term phase 3 trials that have been initiated in PKU adults, and that these trials are also extending down into PKU adolescents. So in conclusion, there are multiple pharmacologic therapies available which now may be used to either reduce or in some cases potentially eliminate dependence upon dietary Phe restriction. These treatments can dramatically lower the burden of the dietary treatment for individuals with PKU. However, I still think there's significant unmet treatment need remaining, and I'm personally convinced that increasing dietary intact protein will, while maintaining lower blood Phe concentrations, will lead to significantly improved health outcomes. And I propose that further novel treatments will be necessary to pursue that goal. And I thank you for your attention.
Thank you very much, Carrie, for this great presentation. Thank you. And now we have even more than 15 minutes. We have 15 minutes for the discussion. So you all are cordially welcome to submit the questions. And let me check. So there are some questions from Professor Anita MacDonald to Professor Carrie Harding. So Carrie, Anita's question is, do you expect that GNT517 will change the protein tolerance? And what is your prediction? How significantly this Phe tolerance will be changed? What do you think about this?
I think it will. It's hard to predict how much. It's planned to be tested in the phase 3 trial.
Mm-hmm. Okay, short answer. Okay.
Until we do it, I don't think we know.
Anita, any comment?
No, I was just hoping that Carrie knew something that the rest of us didn't know.
No, because the trials that have been done, we kept everything constant. It's only been 28 days. We kept We kept their diet stable. And, you know, with a 60% drop in blood Phe, and in some cases, the individuals dropped from, say, 800 or 900, a couple of cases down to lower than 120. I anticipate that, you know, that would allow us some substantial increase. But until we actually do it, it's hard to make a prediction, right? So.
Okay, should I take the next question for Professor Giszewski? We have from the audience, does high Phe in
Moment, Kirsten, because
sorry.
I don't see him. I just well,
There.
Oh, yes, there it is. Hi.
Oh, okay. I will just repeat the question. It's for Professor Giszewski. Does high Phe in males affect fertility, successful conception, and therefore is good Phe control in males important for family planning as well as females?
As I got understood, it is a question about the preparation for pregnancy.
Yes.
No.
Jurek,
maybe you had some problems with connection. Kirsten's question was, is high Phe level somehow related to problems in fertility in men? In men, not only in women, but in men. In men.
No, I don't found any publication about the Phe control in men, but from the theoretical point of view, phenylalanine in high concentration could have a very negative influence of spermatogenesis in men. As I mentioned in my presentation, there are lots of papers, but they were done rather for infertility programs with several failures, that phenylalanine was in the group of chemicals, hormones, and amino acids, which had a negative influence during these 90 days. So if we plan the pregnancy in this couple when not only women but also her partner has phenylketonuria, it is reasonable to prolong this time with low Phe levels before contraception discontinuing, minimum for 90 days. And then if there is a problem, there's no effect, we should start the normal procedure to diagnose infertility. And nowadays, if you maybe know, the main reason for IVF programs, there is men problems. Nowadays, according to some nanoparticles in our environment, the results of spermiogram is tragic. That is true. So young men, they have very often problem. I'm a little bit involved in spermiograms, and when I see the good result, it is very unique. Nowadays, the good result in spermiogram is I don't know, 1 per 100, maybe.
Okay.
And
Okay. Yeah. So in summary, if we can add here, is that when there is a woman with PKU planning pregnancy, and if there are some problems with getting pregnant, so both her and her partner should be early seen by specialists, because maybe the reason of, let's say, infertility in this couple, even if future mom is trying very hard to be on the strict diet, may lie on the future father's side. So this is something which I which I took from your presentation. Am I right?
Yes, yes, of course. The AMH estimation in women, it is very helpful and very simple. Computerized assisted sperm examination also could shorten the time before conceiving with this very stressful situation, especially for women with a special diet. Not everyone is on newest drugs in phenylketonuria.
Okay, thank you very much.
So, Kirsten, if I may ask some more questions to Kari,
Absolutely.
because there are moment, let me organize myself. Moment. There was moment.
Maria, I asked two, if I can remember them.
Yes, but let me find this. Kari, there is a very interesting question from Anita. Would you worry about very low Phe below 40 with women who are pregnant on pegvalace? How would this affect fetal growth?
Certainly, we worry about it. So there have been 16 pregnancies in the literature in women using pegvaliase. So one thing I want to be clear about, you can't initiate pegvaliase. You know, if a woman comes in and she's newly pregnant and has hyperphe, you can't, say, well, I want to start pegvaliase now and treat this pregnancy because it takes too long to get to therapeutic levels, right? So that case, you've got to do something that day or whatever to get the Phe levels down. So where this has been used is, women who have already been on pegvaliase are in good control and then, decide that they're going to conceive, and they've elected to remain on pegvaliase therapy and stay in control. And so there have been 16 published pregnancies. There have actually been many more than that. I don't have an exact number, but there have been significantly higher number of that in the US of women who have remained on therapy. In the 16 published, there have been some women who've had quite low levels, but again, all of them have been on unrestricted protein. I can't say anything about the quality of the protein because I don't think really that was covered very well in any of the publications. The data that are there, the fetal growth has been fine, and the head circumferences have all been fine. The pregnancies that I've been involved with, which have been 2 or 3, the babies have all done extremely well. And I have erred on the side of keeping the phenylalanine levels as low as possible rather than the problem we have is if we lower the dose a little, that tends to let the Phe levels jump back up too high, and I'd rather risk staying low and watching the fetal growth as long as we can maintain protein intake. But this absolutely needs to be studied further.
Okay, thank you very much for this answer. And we have a question to you from Professor Renat Blau. What do you think about combination of GNT-517 with sepiapterin as a single formulation?
I would love to ultimately test this possibility since they work by 2 different mechanisms. I mean, obviously the person would have to be sepiapterin responsive, but I think this would be a magnificent approach to try. If they're 2 different drugs and we'll have to convince insurers that it's worth paying for 2 different pharmaceuticals. The other approach is, can we get companies to work together to actually make a formulation of the 2 in one drug? That's potentially possible, but I think this is a wonderful thought.
Great. And there was again a question to you from Anita. When do you think sepiapterin will be available in tablet format?
I don't know. I haven't asked that question. Anya's been involved with the company. I haven't been directly involved with them, so I haven't heard anything. I don't know if Anya knows.
No, I
about
don't
that.
think that this is planned so far, because a new formulation, you know, is means new studies and so on. And I think they now want to earn the money.
Okay. Okay, and it seems that there are some more comments and question to Jurek. So I will read a comment from Francois Maillot. I remember a poster in a meeting more than 10 years ago in which the authors showed poor spermatograms in 2 males with PKU, so exactly what we were discussing before, recovering with a decrease in Phe levels. So after decreasing the Phe levels, it seems that these men became fertile again. And the authors also mentioned that phenylalanine was seen as a potential contraceptive in men a long time ago. So, as we can see, this was only an information from a poster presentation, so probably more studies are needed. And Jurek, there is another question to you from Professor Anita MacDonald. You just share with us during your presentation this publication from the summer this year by Horka, talking about some fluctuation in phenylalanine concentration in the course of the cycle. And Anita's question is, does high Phe, no, this is not this one, moment. The question, Anita's question is, Jerzy, you show the study that suggested that menstruation did not have large differences on blood Phe, but could menstruation have more impact on some individuals than others? Probably it is more a question to us dealing with PKU than to gynecologists, but what do you think? Is it possible that some women may be more sensitive to the phenylalanine fluctuations during menstrual cycles? What do you think?
Small differences between Phe levels in cycle phases after menstruation, you should know that about 60% of this blood with the active layer of endometrium is reabsorbing. So maybe this effect has some influence on metabolic parameters with phenylalanine inside. So this is very special moment of the cycle when the cycle ends without pregnancy. This whole active endometrium goes out, and with some little blood, there are very important changes in coagulation process. And then 60% of this menstruation is reabsorbed again. So maybe this is the reason why after menstruation, the Phe level goes a little bit up. And then at the beginning of the second phase, luteal phase, the phenylalanine goes a little bit down. The differences among patients are possible because every woman has very special menstruations with volume, with the duration, and it can be very individual, make some reaction in metabolic side, I think.
Yeah, this is a very good, very gynecological point of view answer. And probably it may be also related to some individual variability in metabolic stability during this during the different times of cycles. Anita, are you satisfied with this answer, or would you like to add something? What do you think? There are no data on this.
No, there's very little data, and the little work that's been done is on a small number of patients. So it would be good to monitor this on a wider number of patients, particularly with classical PKU, just to see the impact, because it's a concern of all women. And so I think the more data we have, the better.
Absolutely. And we have one, probably the last question from the whole audience. It is also to you, Carrie. Consider, or maybe also to Anya, considering sepiapterin pharmacokinetics achieving best distribution in hours, take the full dose divided twice a day should be considered. What do you think?
Carrie?
Carrie?
I have only prescribed it a couple of times. I'll tell you, the practical thing is there's so much of it that has to be given. It's easier for my patients to split it. Pharmacokinetically, I don't know if it makes any difference, but Anya might have better information about that. The half-life is pretty long for both cypropirin and sepiapterin. The sepiapterin tends to peak later than cypropirin does in blood, so I think I don't know if it really makes that much of a difference, but practically speaking, it's only been 3 patients so far that I've used it in the last, 3 months. But there's so much powder that they have to take that they've been splitting it mostly, is what I've been seeing. But Anya has way more experience than I do.
Yes, there is always, you do the PK analysis, and there are clear rules what half-life then supports once a day dosage. And this was true for cypropirin. So I personally published this data, but I gave it twice because I'm sure that with a half-life of 11 hours, after 11 hours you have half, after 22 hours you have 25% of the substance. So I never understood why it shouldn't be better to give it every 12 hours. And indeed, what you say, Carrie, the amount of powder helps very much to give it twice a day. Of course, we cannot say this in public because it's not the label.
Okay. Thank you very much. And looking at the time, I think this is a time to close the discussion, and we will give the floor to Professor Nenad Blau.
Yeah, indeed. It was a very good discussion, and I would like to thank all the speakers for the excellent presentations, and also for the audience for a couple of questions. Honestly, I was expecting a little bit more, but as far as I understand, the webinar was recorded, and you will have the possibility to see the information later. With regard to certificates, as an attendee, you need to fill out the evaluation form in order to be able to receive the certificate and credit points. And this can be downloaded from the address, the URL that you see here, elearningmediaacademy.com. Trying to get to the next slide. Yes. On-demand webinar, the recorded webinar will be available, as already mentioned, starting from November 14 on the Media e-learning platform. And credit points will be available for on-demand session until the 7th of May, 2026. One more time to remind you that there are 4 recorded podcasts launched from between June and October 2025. And again, if you go to the website of Medea, medeaacademy.com, events PKU /2025, you can access all these postcards. And the next slide, you can follow Medea on social media. You can see different options that you have, YouTube and so on. And I would like to thank you for attending the live webinar. Please fill, one more time, please fill the post-webinar survey. The survey will be open automatically once the webinar ends. So with this, I believe this is the last slide. So thank you one more time to the speaker and to the audience, and hope you enjoyed the webinar. Take care. Goodbye.